The overall goal of this procedure is to compare the relative affinities of protein-binding partners for Rho-family GTPases under carefully controlled nucleotide loading conditions. This is accomplished by first purifying putative competitive binding partners, each labeled with the same tag, for example, green fluorescent protein, or GFP. The second step is to purify the GTPase of interest, for example, RAC1, and load with the appropriate nucleotide to achieve the desired GTPase signaling state. Next, the nucleotide-loaded GTPase is incubated with a fixed amount of one binding competitor and increasing amounts of the other binding competitor to achieve a titration binding curve. The final step is to resolve proteins bound to immobilized GTPase by Western blot and probe the membrane for the GFP tag that is shared between the two binding partners. Ultimately, the Western blot is used to identify the relative concentrations at which similar amounts of each GFP-tagged binding partner is captured by the GTPase.
The main advantage of this technique over existing methods, like co-immunoprecipitation, is that the protein binding properties of GTPases are dictated by the bound nucleotide. In the cell, the bound nucleotide is labile, making interpretation of protein binding data difficult.
Begin this procedure with purification of GST-tagged GTPase and expression of GTPase-binding proteins, as detailed in the text protocol. Rinse the flasks of transfected cells in phosphate-buffered saline, or PBS, and drain the flask for 5 minutes.